Hello. So we will continue our coverage of sampling methods, specifically the active sampling methods. So we will review first the filtration methods. These methods involve pumping air through a sterile absorbing substance and transforming the filtered microbe onto the appropriate culture medium. So this is sort of a schematics. We have the air being pulled from this end, and we have the filter medium here with appropriate media, and we have the air being pulled through, and the particles, which will be microbes in our case, they will be absorbed or filtered out in this media, and then this sample can be collected. The collected sample can then be analyzed either with microscopy methods or culture methods. Bioaerosols that are captured generally tend to remain viable, or the DNA may be directly extracted from filters for downstream molecular biological analysis. The particles are generally attracted by forces. This could be inertial, diffusion, and electrostatic. Mostly our filters will be having sort of a pump attached at this end. The pump will draw out air. So it's kind of like a air suction force being applied, and the particle passes through the media. Different number of filters can be used. For example, depending on the application, cellulose acetate, nylon, gelatin membranes, glass fiber, and polycarbonate filters can be used. Filters can also be made with fibrous or granular materials like sand, which is then used together with some cotton plugs, and alginate wool, which could be used in phosphate buffer. The pore size and the filter material can be chosen based on the requirement. The flow rates may also be varied, and generally the flow rates in the range 300 to 1000 liter per minute are quite commonly used. These filters have a high collection efficiency and also a good amount of survival rate of the microbe. Typically, 95% of particles that may be as small as 0.035 micrometers in diameter can be captured. So we can go to a very fine particle diameter size using this method. Therefore, this kind of method is applicable to a wide variety of bioaerosols, from bacteria, fungi, and even viruses. The efficiency of the method, as we can gauge, depends or varies with the sampling time and the relative humidity. For this kind of method is quite widely adopted for simplicity, because we only need to have a filter and a filtering mechanism and a suction pump, and then we will be able to filter the air onto the filter media. So it is widely adapted for simplicity, for long duration sampling, and also the economy. So one would need probably electricity cost, but otherwise the sampling assembly can be kept and can be used for a long period of time, so that sufficient bioaerosols are filtered into the media. Because of the lower size range that we can obtain through this filtration, because of the lower size of the particles that can be trapped in this filtration, it is possible to detect the very small aerosols to the size that can create the respiratory implications for humans. So we know the respiratory fraction can be there, and we can quantify, if it's a single filter, we may be able to quantify the size ranges that were trapped in the filter. But if we are able to use a series of filters, potentially we can also put them in size bins, that so many particles are in this size range, so many particles are in this size range. So we can in general get a gauge of how many are in a particular size range. Filtration method have some drawbacks in that, or some caveats that need to be taken care, in that the sampler suffers from the impaction in overloading colonies when we have a lot of colonies. The desiccation generally kills the microorganisms that may have survived during the collection. So we need to somehow prevent the desiccation of the microorganisms. In particular, gram-negative bacteria, they may not tolerate these kind of stresses. So for this case, probably hydrated filters, or filters that may prevent this desiccation or minimize the desiccation, may be used to prevent the underestimation. Because if we are trying to analyze viable microbes, and the desiccation when on the filter paper has killed the microbes and not in the air, we are getting a lower estimate, because if we had 100 microbes, desiccation killed 50, we are only measuring 50 as viable, whereas the actual number was 100. So desiccation may kill the microbes that survived during collection. So somehow, if we can have hydrated filters, they may be used for to avoid this issue and prevent the underestimation. Sometimes higher flow rates and longer sampling duration can result in loss of spore viability. Also, filtration is not adopted for fragile bacteria. The next is the impingement methods. So impingers collect microbes in air samples into a chosen collection fluid, which would be a sterile solution of water, surfactant, mineral or glycerol salt. A high volume pump generally draws the air through a special all glass impinger at a flow rate of approximately 12 liters per minute. The phenomena involves sucking the air through a narrow inlet and impinging on a collection medium as soon as the air strikes the liquid. So here the liquid is somewhere down and the air is sucked. So this is just a conceptual diagram. So as the air strikes the liquid, our bioaerosol of interest sort of goes and gets collected on the medium. So these black dots are basically coming through the bubbles into the liquid, and they may be there or maybe on the surface, and then the air goes out. So this all glass impingers are also abbreviated as AGIs. So air glass impingers or AGI, they have a pre-established distance between the inlet jet to the base of the impinger. So the term AGI4 means distance to the base is 4 millimeters. AGI30 implies the distance is 30 millimeters. So this number basically tells the distance between the inlet jet and the base. So AGI4 is an efficient particle collector, but here this bacteria may go under greater physical stress due to the shorter distance between the jet and the bottom of the impingers. It's very close, 4 millimeters. So if we are sucking through or we are passing a fast jet stream, there is a physical stress involved. The recovery of viable microbes is more likely with the AGI30, which is a relatively inexpensive and efficient sampler that utilizes a low flow rate to bubble air through the liquid matrix. This is how a impinger might look like. So we have the air coming through the inlet, then we have the collection vessel here, and here we have the tangential nozzle. So here the air is basically impinged on the collection liquid and the biological material is entrapped. Then this air goes up and goes out through the outlet, and this is how it may look in a lab. So impingement is a technique similar to impaction, except that the organisms are collected into a liquid medium. So once this is a multi-stage liquid impinger. After sampling, the aliquots are prepared and run through a suitable media to count the viable microorganisms. So once our collection is done, we can do the further analysis. So the inlet characteristics of the sampler will influence the collection of the bioaerosols and the recovery efficiency of the bioaerosol, quite similar to the impaction methods. The recovery of microbes may vary with flow rate, wind velocity, and also the orientation. Generally, higher flow rate may tend to reduce the recovery. In such methods, generally or sometimes isotonic or buffer medium is added to avoid the osmotic stresses on microorganisms during the sample collection. These impingers have one could say reasonable collection efficiency. They generally have a collection efficiency of 70% at 0.5 micrometer and 90% at 1 micrometer. The entrapment of air through the liquid makes dispersal of particles in the liquid, followed by the quantitative measurement through serial dilution, for example. Effective recoveries in mineral oil and glycerol have been observed in the impingement methods. So, as compared with the previous active sampling approach, the method can generally be effective in the overcoming of the problem of desiccation. So, we may be able to retain more viable bioaerosols in the medium. The process can allow for different media to be inoculated from the same sample and may also permit greater sampling times than the 30 minutes that are generally used, but one can in principle use this method for a longer time. From the collected material, separate aliquots may be analyzed for culture and non-culture based examination. The method has been shown to be efficient for collecting a diverse range of particle sizes, but sometimes less effective for hydrophobic bacteria such as bacillus and some mold spores in water. The evaporation of impinger liquid occurs in longer sample runs. Sometimes this may happen, causing sample loss and desiccation. But if the experimental run is not very long, this may not happen, but if it is very long, this is a possibility. The collection fluid then needs to be refreshed to minimize these losses. Glass tubes that are used for collecting bioaerosols can also break due to handling errors, and due to the low flow rate, impingers can have sometimes low capture rates. It has said that sometimes virus-sized particles may not be that well collected. The next method is centrifugation methods. In this method, bioparticles from air are passed along a tube that rotates rapidly on its long axis. So, some kinds of centrifugal air samplers are portable and battery powered, and they can be taken anywhere and can be used quite user friendly manner, in a user friendly manner. This portable rotor centrifugal sampler could represent a large cylindrical torch with an open-ended drum at one end that has impeller blades with plastic strips coated with the culture medium. So when the centrifugation is happening, the bacteria or the microbes may attach to the culture medium. So with an inflow air flow rate of about 50 liters per minute, the suspended particles are impacted upon the culture medium. The sampler itself is a bit easy to be transported and can be used for longer durations. It is sometimes said that this is less efficient than methods such as slit samplers in detecting particles below 5 micrometer in diameter. Moreover, the size of particles that are coming in the air being sampled cannot be accurately controlled. This is a sketch of how a sampler could look like. The principle is basically of impaction and drawing the bioaerosols onto an agar media strip. The centrifugal motion in this case can take air up to about 100 liters per minute. The next type of samplers are the cyclone samplers. They are advantageous in that they address some of the shortcomings of impaction and impingement. In cyclone samplers, microorganisms are captured into sometimes liquid medium using swirling air and centrifugal force. So this is how such a sampler may look like, and this is our conceptual diagram. So the microbes containing air is drawn into the cyclone, and then the air takes a number of turns and impacts on the side of the cyclone. So when these particles are striking the corner or the edges of the cyclone, they will lose the energy and then come down where they are collected into a medium, and the air which is now free of these particles can go out of the cyclone sampler, and this is the place where we can collect our microbes into a medium. So the method is stable, but it may be susceptible to particle bounce and re-entrainment. Some of these particles can move up again. Sometimes water can be used as a collection medium, and maybe sometimes it is good for this method is good for collecting the gram-negative bacteria. Then the next technique is electrostatic precipitation. The technique is based on the particle precipitation by electrical force and the electrical force being applied to the charged particles. This method is quite similar or essentially the same to the electrostatic precipitation method used in cleaning flue gas, industrial flue gas from particulate matter. The particles are charged at the inlet, followed by exposure to electric field inside the chamber. So let us say these are the conceptually again, these are our particle containing air coming in, and we have the metal rods that are basically charging the inlet particles. So when they go inside the sampler, we have the opposite sort of field. So if the particles are negatively charged now, they will be attracted towards this positively charged plate. So this is air containing bioaerosols. The charges are impacted through these metal rods, the yellow color rods. Then the particles move in together with the air, and now since there is an electric field, the positive metal positively charged plate will attract the negatively charged ion, and then the particle basically negatively charged particle, and the particle will go and deposit to these metal plates. So this is our cross-sectional migration, and then the particles are deposited over charged plates, and from here they can be extracted for analysis. This method minimizes unwanted stresses on samples and the collection efficiency is mainly controlled by the parameters such as voltage applied across these plates, the flow rates, the dimensions of the precipitators, and the particle charging levels. Sometimes very low power may be needed for charging of precipitators as related to the collection of bioaerosols, but the electrobiological properties of the microbes can vary considerably. So some of these microbes, like certain bacteria, may become non-viable during sampling because they may not survive this flow through charging or this flow through a charged field. The next method is thermal precipitation. This is one of the old sampling techniques, and it is based on the thermophoresis principle. Basically, the movement of particles from a high temperature region to a low temperature region. So in this method, the air laden bioaerosol is passed through a narrow channel. So this may be conceptually again, this may be the air with the bioaerosols going through a narrow channel containing a temperature gradient perpendicular to the airflow. So we have hot plate here and a cold plate here. So airborne particles will tend to move away from the hot surface around, maybe sometimes they are maintained at around 125 Celsius, towards the cooler surface and then depositing over it. So now the particles are moving, and of course some gravity may be involved, and this temperature and the temperature gradient is there. So the particle will go and deposit towards the cold plate, a phenomenon known as thermophoretic motion. The collecting cold surface is cooled by a circulating water heat exchanger. So in this region, we may have cold water being circulated so that this cold temperature is maintained and the particle may continue to be deposited. Microscopic examination can be done of collected matter can be done in glass microscopic cover slips that are used as the cooler collection surface. So our collection surface itself may allow for examination of the microbes, or can be collected over filter paper from which one can transfer to agar plates for further analysis. The collection efficiencies can be very high for small particles in the range 0.01 micrometer to 5 micrometer. The method is also used to determine the particle size distributions. One part to note is that collection efficiency for smaller particles is high, but the collection rate may be low compared to other samplers because the rate is around 7 cm³ per minute to 1 liter per minute. The next technique is condensation. Condensation technique consists of a number of parts attached together, such as vacuum pump, humidifier, a heating source, a liquid source, amplifier, a cooling source, and a biomass spectroscopy system. So in this type of mechanism, air is drawn in through a vacuum pump into a humidifier with a heating source evaporating the liquid to create a humid environment. So the relative humidity could be 90% or even higher, and temperature higher than 35 Celsius but lower than 45, so that the heating happens but the microbes are not deactivated. So this kind of temperature is maintained. The air sample is passed into this amplifier with cooling surface via vacuum action, and the temperature then is reduced to temperature as low as 10 degrees Celsius. Due to this low temperature, the air of the amplifier is subjected to condensation with these vapors. Sampled bioaerosol will act as a condensation nuclei, and once the vapors are condensing on these nuclei, the particle grows or amplifies, and the viability of the microorganism is also maintained. The particle size amplification due to this condensation for the bioaerosols can occur at the order of nanometer and submicron scale and help in the detection efficiency increase because the particles have grown in size. So in summary, we have seen several of the methods. One was this in the previous lecture, sort of a passive sampler method in which the bioaerosols were deposited in a agar plate and deposition was happening under gravity. To other methods such as electrostatic precipitation, movement in an electric field, thermal precipitation, movement of particles in the thermal regime, the impactors. So particles are moving in, being impacted, then moving in, being impacted. So multi-stage impaction, and then they are collected. Cyclone, filtration, and impingement. So in summary, a variety of active sampling methods and devices have been explored, ranging from impactors, filtration, impingers, centrifugation, cyclone samplers, electrostatic and thermal precipitators, and condensation methods.